Let's turn on my sound. That will help. All right. So I told you we'd start off with this today just to refresh your memory on what we did yesterday. Hopefully you went home and drew this out a little bit. All right, so remember that water is going to be split on the side of the thylakoid membrane facing the thylakoid space, and we do split that into hydrogens, protons, and electrons. Where did we say that hydrogens, electrons go?
Chlorophyll?
To replace those lost by chlorophyll. And what is this center part called? Where is that chlorophyll A? The center of a photosystem. Yeah, that's the reaction center. So those electrons are going to replenish those electrons that were lost by chlorophyll A in the reaction center of photosystem 2. Where are hydrogen's protons going to go? Well, notice here, that's those two H pluses. They just come right off into the thylakoid space, and then oxygen is going to come off as 1/2 O2 for us to breathe. Okay, so a photon of light strikes a pigment, and that photon, that light energy, is going to sort of bounce around from pigment to pigment until it gets to that reaction center, where it's going to knock 2 electrons loose and raise them to a very excited state. Now remember, those electrons are not very stable at that excited state, and so they will gradually start to lose some of that energy that they gained. And as they lose some of that energy, we are going to use the energy that they're losing to actively transport H pluses from the stroma into the thylakoid space. And then once that H plus concentration is large enough in that thylakoid space, then the gate on this ATP synthase channel protein will open and H pluses will rush through. And as they rush through, they're going to cause this little turbine part at the bottom to spin, and that's going to provide enough energy to add a phosphate group onto ADP to make ATP. Now notice that ATP is being made out here in the stroma, which is convenient because ATP is going to be needed for the Calvin cycle, which we'll learn about Monday, and that takes place in the stroma. And so the two products of the light reactions that you need for the Calvin cycle are ATP and NADPH, and those are both going to be made in the stroma. Okay, so we got to start today talking about a term that's kind of related to chemiosmosis, but it's a little different. And this is called photophosphorylation. Okay, so photo we know means light. Phosphorylation, we know that has something to do with phosphate groups, and so we're going to see how we tie this together. Okay, so phosphorylation, the addition of a phosphate group to ADP in order to make ATP. So we're going to talk about two different types of phosphorylation between photosynthesis and respiration. So in photosynthesis, since we know that this is so dependent on light in order to make ATP, it's always going to be photophosphorylation. But when we do cellular respiration, we have two kinds in that are not related to light, which are substrate level phosphorylation and oxidative phosphorylation. So we'll get to those in the next unit. But let's figure out what this means. Basically, when we're talking about photophosphorylation, we're basically saying, why is light necessary to eventually make ATP? In other words, if I didn't have light, why am I not going to be able to make any ATP? What is light really needed for here?
Make the electrons.
Exactly. So let's think of like a cause and effect, right? That if we don't have light, then we don't have anything to excite the electrons. Okay? And if we can't excite the electrons, then they can't slowly come down from that excited state. And what are we going to use the energy that they're losing to do?
Put hydrogens in.
Right, you're going to use it for active transport to put hydrogens into the thylakoid space. Okay, it's only when that concentration of hydrogens in that thylakoid space gets large enough that that ATP synthase channel can open and hydrogens can rush through, and you can add a phosphate group to ADP to make ATP. So all that to say, you know how I am about this leads to this, which causes this, which causes this. You know that's how I roll, okay? That's really all photophosphorylation is saying, is that you need the sunlight to initially energize those electrons and raise them to an excited state, because when they come down from that excited state, you're going to use that energy that they're losing to actively transport H pluses from the stroma into the thylakoid space. And then once that concentration is large enough, we can open up ATP synthase, and then H pluses can rush through, and that will provide enough energy to add a phosphate group onto ADP to make ATP. So yes, I'm writing that out in your notes, but understand, I want you to understand the light reactions well enough, okay, that you don't memorize this. If you understand step by step what's happening in the light reactions, which I think you will, if you don't already understand it, I think you will understand it better once you go through this poster activity. We've got a poster activity, we've got a short computer lab to do on your computer. It will not take you probably more than 15 or 20 minutes. And then you'll start on your review. So I'm trying to break it up a little bit today. I didn't want to do a ton of notes, but we got to get through this packet. And then we'll do the poster activity where you'll record yourselves and upload it, short computer lab, and then you can work on your review. All right, so that's kind of just an aside, like underneath chemiosmosis. Make sure everybody's got that. All right, so now that we've talked about chemiosmosis, now we can talk about those electrons eventually landing in the reaction center of photosystem 1. So if I were to ask you to take me through the steps of the light reactions or the steps of non-cyclic electron flow, which is this, you know, you got to do things in the right order. You can't talk about the electrons and what they're going to do in photosystem 1 until you have talked about chemiosmosis, because chemiosmosis happens during that first electron transport chain. It doesn't make sense if you go back and talk about that later. So you've got to talk about chemiosmosis when the electrons get, or once they have come through that first electron transport chain. So they're gonna land in that reaction center, another photon of light is gonna come in, and then bounce around from pigment to pigment until it gets to that reaction center, and again, excite those electrons there. Now, yes, we're gonna go through step by step, but I don't want you to think, oh, why is she making us write this? It's just the same thing that happened before. You can look at your diagram and see it's not the same thing that happened before. We have different proteins in the second electron transport chain, and the second electron transport chain is much shorter than the first. And that is significant. So yes, some of this will seem like a repeat, but I promise not all of it will be. So that photon of light striking or getting to the electrons in the reaction center is going to excite those electrons, raise them to a higher energy level where they'll be accepted by the primary electron acceptor. Okay? Electrons will then enter a second electron transport chain, which is much shorter than the first one, and will include the protein ferredoxin. Okay, so my people who've had chemistry, when you see that prefix on the last word there, what element do we think of? That's a Jeopardy question. No? Come on. Some of you have Tedder. I know you have learned this, but that was like 2 years ago.
Yeah,
So
it was 10th grade.
anybody want to take a guess? The ferro part?
Is it iron?
It is iron. There you go. Did you have Miss Palmer or Mr. Tedder?
Mr. Tedder.
Oh, there we go.
Yeah, I could stand it.
I know. However you felt about him, he did teach you some chemistry. And you can also get that, right? Because iron's symbol on the periodic table is Fe. It's because it's from the lab. Anyway, I'm not trying to get into a bunch of chemistry in here, but I do like my trivia. All right. Primary electron acceptor is going to transfer these energized electrons to ferredoxin. So this is kind of what this second electron transport chain looks like. Okay, we have those excited electrons, and they're going to get passed to ferredoxin, and then they're going to get passed to an enzyme called NADP plus reductase. But look at where my hands are. Are they as low as when I went through the first electron transport chain? No, they're about halfway down. So because this second electron transport chain is shorter, the electrons that you get at the end of it are going to be much, much higher in energy than the electrons by the end of the first chain. And that's significant because we like high energy electrons. We actually have specialized molecules that like to carry high energy electrons. So that's the significance of that. No chemiosmosis occurs in this second electron transport chain, and so you do not make any ATP here. Okay, so ATP is only made during the first electron transport chain. Oh, I'm not going through this whole diagram. I'm just looking at this app. So we kind of picked up today with those electrons landing in the reaction center of photosystem 1, and then another photon of light is going to come in. It's going to bounce around from pigment to pigment. It's going to knock 2 electrons loose. These are the same electrons that landed here. okay? So I don't have to have a replenishment source of electrons from photosystem 1, because the electrons I'm exciting are the ones that came down that chain anyway. And so they raise them to a higher energy level, we're gonna pass them to ferredoxin, then we're gonna pass an enzyme called NADP plus reductase. And I always think of NADP plus reductase as kind of a chef, okay? It's gonna take some protons, it's gonna take some electrons, it's gonna take some NADP, okay? And then we're gonna combine them all together to make a compound called NADPH. And remember, when we talked about oxidation and reduction, we said we were gonna track electrons by tracking hydrogens, okay? And specifically by tracking hydrogen's electrons, because notice how that chain is shorter. Those electrons have not come down all the way from their excited state, so they are not just ordinary electrons, they are high energy electrons. And that is what is going to get combined with NADP plus to make NADPH. So that H there, yes, includes a proton and electron, but those are not going to be just ordinary electrons. Those are high energy electrons. Okay. So electrons are transferred from ferredoxin to NADP plus, and this reaction is catalyzed by NADP plus reductase. So again, I kind of think of NADP plus reductase as kind of like a chef who's going to throw a bunch of things together to make NADPH, which is another one of your major products from the light reactions. So it's going to involve taking those electrons that just were handed to NADP plus reductase from ferredoxin. Basically, where this is, if you look at this previous picture, if we look at this, no, not that one. Go back. Okay. If we look at this previous picture, this is NADP plus reductase. So ferredoxin is going to pass the electrons to it. The NADP plus and H pluses are out here in the stroma. Can you tell me where those H pluses came from?
The windmill.
There you go. They came from down there. Exactly. So the H pluses that are here came from there. And in your poster activity, you'll be showing that. Okay? So electrons are coming from the chain, protons are coming from down there, and NADP plus is coming from the Calvin cycle because it's something you produce from the Calvin cycle, and then that can be a reactant here. We're not saying the light reactions are a cycle, because they're not, but the light reactions in the Calvin cycle sort of cycle back and forth, at least some reusable things. Good. Nope. Nope, that's where we are, right? Okay. So this reaction is catalyzed by the enzyme NADP plus reductase. 2 hydrogens from the stroma are needed. Those are the ones that came through ATP synthase. And this forms NADPH, which is an electron carrier. But NADPH doesn't just want to carry ordinary electrons, it wants to carry high energy electrons. And so that's why how we know they're high energy is because they didn't come all the way down from that excited state. And so something that my students usually have to get used to is we know with ATP, we know that the energy is stored in the bonds between the phosphate groups. In NADPH, it's really just called an electron carrier. It doesn't store energy the same way that ATP does. It stores electrons that have a lot of energy. It stores high energy electrons, but we say, ATP is energy storage. We can break a phosphate group off to yield energy for the cell. NADPH, it's got that H on the end. That means it's got some high energy electrons. So they function a little differently from each other. All right. So when I'm going from NADP plus to NADPH, which substance got oxidized or reduced?
NADP plus got reduced.
Awesome. NADP plus gets reduced to become NADPH. So you got to remember that when we're talking about oxidation and reduction, we're always dealing with hydrogens or the electrons from hydrogen. What does ATP stand for? Well, adenosine triphosphate, because it's got the sugar on it. But understand, there's no H in there. I'm not saying ATP doesn't have hydrogen. I'm sure it does. But when we talk about what gets oxidized, what gets reduced, ATP is not involved in that whatsoever. ADP is never the answer. If I ask you what gets oxidized, what's reduced, it's never ATP or ADP or anything like that. Okay? It's just not. It really is just tracking the hydrogens. For some reason, people want to put ATP down, and I'm like, that has nothing to do with it. Where to next? The ATP and the NADPH produced during this process will be used in the Calvin cycle. And as I mentioned, the Calvin cycle is going to take place in the stroma. And ATP will be an energy source. NADPH will be your source of high energy electrons. For the Calvin cycle. I don't have my notes in front of me, so I don't know what's next. That little analogy diagram, which is kind of silly, but some people it works. No, this is not the kind of detail you need to know this in, but I do kind of like the analogy of it, that we have a photon that is exciting that electron and raising it to a higher energy level, and then it's saying the mill makes ATP. Well, this is the first electron transport chain, and we know that's when what process happens? Starts with C. Chemiosmosis, right? That's when chemiosmosis is going to occur. And then that electron is going to land here in photosystem 1, and then another photon is going to come in, and it's going to excite that electron and raise it up to a higher energy level. And you have a much shorter electron transport chain, which isn't really shown here, but it shows the electrons in a bucket. And so that's kind of the way I think about NADPH, is we call it an electron carrier. And so that's why they're showing it in a bucket, because it's literally an electron carrier. So yeah, that's all I want to say about that. All right. Let's do our ins, outs, and wheres. Because even though this is not all you need to know about this, it is some of what you need to know. So I always like to summarize a process this way. Let's see. What's going in? Tell me some things going into this process.
Photons.
Photons, good. Light energy. All right, another major N.
NADP plus.
I'll take that. That's a minor N, but that's still an N. I have one more minor in, one more major in, whichever one you want to give me. You go along with what you just said for the second one.
Hydrogens, because they come
No,
in.
they're gonna be there anyway. Think along those same lines though. There's something else that's a precursor to what we're gonna make here.
Water,
Water you need, that's a major in. But now we just have one more minor in. What do I make during chemiosmosis?
ATP.
Where does it come from? It comes from ADP. And so that ADP, these are both gonna come, and really it's ADP plus P. These are gonna come from the Calvin cycle. So they will be produced from the Calvin cycle, and then they will go in to the light reactions. All right, okay, let's switch to out. Three things. These are all big outs. Well, I know you can look at the ins and tell me at least two of the three outs.
NADPH.
And ADPH is one.
Hydrogen's leaving the cell.
That's negligible.
ATP.
ATP is another one. And then the whole reason you're alive right now,
Oxygen.
oxygen. Okay? Where is going to be on the thylakoid membranes in the chloroplast? I'm just going to say on the thylakoid membrane because you should know that those are in a chloroplast. So I like doing that after we get through a major process. I think it helps. It's not that I want you to memorize. I kind of want you to be able to, like, I can see some of you trying to think through those diagrams in your head, and that's what I kind of want you to do, as opposed to just saying, okay, I'm going to cover this up with my hand, and I'm going to memorize it. You're not going to know it long term if you do that. Try to think through, if you're visual, try to think through those diagrams in your head and remember what you made at different points. I'm not going through that again. All right, last little topic for today. Is cyclic versus non-cyclic electron flow. Now notice there's not many slides in this part. So understand what we just got done doing is non-cyclic electron flow. Sometimes they call it the Z scheme, okay? But cyclic electron flow is something, it's like a little detour that can happen every so often, and there's a reason that it happens. It's just kind of an alternate path. I don't even want to call it that. You'll see. Okay, so non-cyclic electron flow is what we did yesterday and today in notes. Sometimes that's called the Z scheme. I don't really call it that. It's using light energy to produce ATP and NADPH. So of course this is non-cyclic because what you make at the end here doesn't come back and go into the beginning. Now don't confuse that. Notice this little part of the picture here. There are things that cycle back and forth between the light reactions and the Calvin cycle, and that's your NADP plus, your ADP plus P, your ATP, and your NADPH. Those things cycle back and forth between the light reactions and the Calvin cycle. But this thing itself is not a cycle, okay? So, So cyclic electron flow is going to involve photosystem 1, but not photosystem 2. It is a way to generate extra ATP, but it will not make any additional NADPH or O2. It's going to be basically like repeating part of that non-cyclic electron flow diagram. It'd be like if you had downloaded an entire album or CD or whatever we call it now. I know we don't use CDs because we have digital music, but let's say it was a whole album. You're like, oh, this rocks. I got to download the whole thing. And there was one particular song you liked, and you just kept going back and playing that song on repeat, on repeat, on repeat. That's essentially what this is. It is repeating part of the light reactions over and over and over again to make additional ATP, because the Calvin cycle that's going to come next is going to require more ATP than NADPH. Okay, so non-cyclic electron flow produces roughly equal amounts of ATP and NADPH. So if we just do that process that we just finished talking about, you're going to make roughly equal amounts of ATP and NADPH. But the problem is, Calvin cycle requires more ATP than it does NADPH. So if I don't take a chance every once in a while to just go ahead and make more ATP, then eventually this process is going to lag behind, because it's going to have enough NADPH but not enough ATP, and it needs both. In fact, of course, it needs more ATP than it does NADPH. And so that's why every so often, instead of doing the entire light reactions diagram, it'll switch to this cyclic electron flow and just do a couple of the steps on repeat to generate the extra ATP that you need for the Calvin cycle. So the Calvin cycle is going to just produce some ATP to make that little bit of extra that you need for that Calvin cycle. And this is showing you, I think I have a picture on the next slide that's bigger. All right. Let's see what we recognize. There's ferredoxin. Ferredoxin was over there. Okay, it's a protein. Can proteins move around in the membrane? Yep. Can phospholipids move around the membrane? Remember we said the membrane's fluid, and that means that phospholipids and proteins can move around pretty freely in the membrane. So the fact that ferredoxin used to be over there and now it's here is not a big deal. And so basically, yes, the photon of light comes in, strikes a pigment in photosystem 1, it excites those electrons, it still passes those electrons to ferredoxin, but now notice what ferredoxin is doing. Ferredoxin is going to move over here, and it's going to pass its electrons to cytochrome complex and then plastocyanin. And so think about how that looks, okay? Photosystem 1, electrons get excited to here. We still pass to ferredoxin, but now instead of just passing to NADP plus reductase, which would make NADPH, we don't need more NADPH. We need more ATP. So we basically got to repeat the chemiosmosis step, okay? So when ferredoxin gets the electrons, it passes them to cytochrome complex and then passes them to plastocyanin. And as those electrons lose that energy, what are we going to use that energy for? What do we do with that energy the electrons are losing? Yeah, I'm going to actively transport those hydrogens into the thylakoid space. I'm going to build up an H plus gradient. And when that gradient is big enough, then ATP synthase is going to open and the H pluses are going to rush through, and I'm going to make some ATP. So to me, this is very much like keeping a song or a couple songs on repeat, because you are just repeating certain steps You're still using ferredoxin, but it is passing to cytochrome and passing plastocyanin. So those electrons do come all the way down from their excited state, which means I can do active transport. That's just what we just went through. So this is just detailing that in notes form. And then that should be our last slide for today. Okay, so when electrons in photosystem 1 get excited, primary electron acceptor still passes them to ferredoxin just like it would have before, but instead of passing the electrons to NADP plus reductase, ferredoxin passes them to cytochrome complex and then to plastocyanin. And then as those electrons lose energy, as they go from ferredoxin to cytochrome complex to plastocyanin, the energy that they gave up is used to actively transport H pluses into the thylakoid space to build up a proton gradient so that we can eventually make ATP. I'm just gonna go in my prep room real quick and get what I need for posters. Okay, let me see how many of these I have. There. That goes with it. 1, 2, 3, 4. Okay, so like yesterday, we're gonna work in like 7 pairs of 2, but we can have a group of 3. So I've got to give you your sheet first. Let me end my recording and I can give you your direction sheet.